So I'm going to talk about the structures that we haven't seen so far in the schematic diagram of the basal ganglia, the subthalamic nucleus. And I'm going to present within this 25 minutes-ish all the different steps that led us to propose that this nucleus that is really tiny in the rat's brain could be a very interesting target, possibly for the treatment of addiction and other impulse control disorders, in fact. So, first of all, for those of you who were attending the session on DBS this morning, you've heard of the subthalamic nucleus as a target for Parkinson's disease treatment. And that's what mostly people within the basal ganglia community know the subthalamic for, and as a motor structure. So it's involved in this motor loop that is illustrated. And it was shown in the 90s that there was an hyperactivity of this structure in the case of a dopaminergic depletion, like in Parkinson's disease. And therefore, lesioning this hyperactive nucleus could restore movement in MPTP monkeys, the Parkinsonian models. And in '93, Ahmed Benazouz in Bordeaux, in France, showed that high-frequency stimulation of the subthalamic nucleus could actually alleviate muscular rigidity in Parkinsonian monkeys. And that's only in '95 that the first paper reporting high-frequency stimulation of the subthalamic nucleus in Parkinsonian patients was published by a French group of Professor Benabid in Grenoble. And since then, now this therapeutical approach has spread all over the world, and you are probably all aware of that successful treatment for Parkinson's disease. But in parallel to that, I was working in the STN of rats, and we had seen that we could actually alleviate some motor signs of behavior in our Parkinsonian rats, but we were also affecting something else. And then that's what led us to look at the non-motor involvement of the subthalamic nucleus. And indeed, as you can see on this diagram, the STN is also involved in a non-motor loop of the basal ganglia, involving direct input from the prefrontal cortex, but also an indirect loop with the nucleus accumbens and the ventral pallidum. All these structures are really well known to be involved in what we call frontal functions, such as attention, the control of inhibition, and motivation. So that's all these aspects of behavior I'm going to screen quickly today. And but we also knew from data in human, it's a case report in '95 published by Trillet and collaborators, that an infarct at the level of STN could induce speech disinhibition and mood exaltation. And also, yeah, there were our first data using STN lesions in rats and showing that it could alleviate akinesia, but induce side effects that were possibly related to a problem of attention. So the next step, and there are, of course, now, since then, there were also cases of Parkinsonian patients subjected to the DBS of STN, for whom mood changes had been reported. Mostly depressive state cases, but one mirthful laugh crisis that was described by Paul Crack in Grenoble. And most interestingly, for the last part of my talk dedicated to motivation, PD patients subjected to subthalamic nucleus high-frequency stimulation gain weight, and it's not just anecdotal, it can be a really impressive gain weight, so I'll come back to that. But the first step was to look at the involvement of subthalamic nucleus in attention. And we've heard during the meeting about the 5-choice serial reaction time task already. And this task allows to assess divided attention in rats, but also we can measure many aspects of impulsive behavior. So I'm going to briefly describe what the rats are supposed to do. They have to start a trial by making a nose poke in the food magazine, and then they have to turn, wait, and detect a brief stimulus light that comes in one of 5 possible holes, and then they have to make a response by making a nose poke in the appropriate hole. If they do so, they get a pellet and they go back to the magazine. Therefore, they initiate the next trial. So it's also involving sustained attention because they constantly start the next trial and have to move on. So, but as you can see on the lower part of the graph, there are many different things we can record in this task. So, for example, if the rat doesn't wait for the light, but goes immediately to make a nose poke in a hole without waiting for the light, that's what we call premature response, and that will be an index of their impulsive behavior. They can, of course, make incorrect response, so they poke in a wrong hole, and they will be punished by a timeout. And they can also omit to respond within 5 seconds. If they didn't make a nose poke, then they're also punished by a timeout. What we also record in the task is a number of what we call perseverative responses. So after they've made their first response in a hole, if they keep going poking in holes, whatever the response was, whether it was correct or incorrect, if they keep poking, then we record that as perseverative responses. And we also measure the number of perseverations they make in the magazine where they collect the rewards. That's an index of their motivation. And as well as the perseverative responses, we also consider that it can be considered as an index of compulsive behavior. So in the next slide, I'm going to show the effect of bilateral excitotoxic lesions of the subthalamic nucleus on the left-hand part of the slide, and effect of bilateral high-frequency stimulation of the STN on the right-hand side. And as you can see, so I didn't show the first part of the results showing accuracy of performance, omissions, correct latencies. So everything is impaired after STN lesions in that task. And, but more specifically for what we wanted to talk today about, the control of impulse control disorders, then I focused on the premature responses, that is the index of impulsivity, and you see that STN lesions increase dramatically the level of premature responses. So these rats can't wait, and they go and poke in holes, they don't wait for the light. And once they start poking in a hole, they keep poking in different holes. So that's why we have this huge level of perseverative responses as well. And once they shift from the holes, they go to the magazine, hoping they get a pellet, and they keep poking, checking if they earn something. So you see that they are highly compulsive, impulsive after STN lesions. Surprisingly enough, when we apply bilateral high-frequency stimulation, we don't see any premature deficit. But we see perseverative responses and also perseverative visits to the magazine. So there are slight differences. Although at that time when we started to work on DBS, we thought that it was mimicking an inactivation of the structure. And you'll see that for some aspects of behavior, it really mimics exactly the same effect as a lesion. Applying DBS does not only inactivate the neurons, and that's been discussed in the session this morning for those who attended it. So, but interestingly, for what we were looking at, I mean, I was working at Trevor Robbins' lab at that time, and he wasn't at all interested in the STN. So, of course, he was assessing many of different lesions in the brain, but not the STN. So then we decided to review what are the other structures inducing the same type of deficits related to impulsivity. And then with Dawn Eagle, we've written this review looking at the control of inhibition system in the rat brain. And for example, the first step was to look at which lesion in the brain induce the same deficits, increasing premature responses and perseverative responses in the 5-choice task. So that's what is circled in pale orange and dark orange on the graph. And you see that we have only 3 structures that share the commonality of both inducing increase on premature and perseverations. It's orbitofrontal cortex, dorsal medial striatum, and the subthalamic nucleus. So right from now on, we have already a disagreement with Trevor because the accumbens is only not doing exactly the same thing. So there is not the same involvement on the control of impulsivity with the accumbens than within these 3 structures. And then, of course, we use different other tasks to assess impulsivity. And so the stop signal reaction time task and the delay discounting task. I'm not going through the results, but I just want to highlight again a very obvious network that comes to induce exactly the same deficits in all these tasks. Those rats, the structures highlighted in red are the results in the stop signal task. So when you lesion the orbitofrontal cortex or the dorsal medial striatum or the STN, those rats can't stop. So once they're engaged in producing a response, they have really high difficulties in stopping. While you see the accumbens doesn't do anything, and when they have the choice between a lever where they would get an immediate small reward versus another lever where they get a large but delayed reward, for once the STN rats can wait. And that's a really surprising effect we've seen. And actually this result has also been seen in the orbitofrontal cortex and the dorsal medial striatum. So to make the story short, the first conclusion is that STN lesions induce high level of premature and perseverative responding in most behavioral tasks, 5-choice, reaction time task, many other tasks we've used. But these STN rats, they're able to wait if there's a bigger reward that's on board in the delay discounting task. And that's similar effect to orbitofrontal lesions. So it questions, of course, the assessment of motivational functions in the delay discounting task and also the effect of STN lesions on motivation. As I said, they were also perseverating towards the magazine in the 5-choice task. But also STN lesions prevent the animals to stop the ongoing action in the stop task, and that's similar to orbitofrontal lesions. So it also questions the role of this hyperdirect pathway that has been highlighted in the recent years, connecting the orbitofrontal cortex to the STN. And this connection really needs to be assessed precisely. So and as I've shown you, there's an obvious circuit for inhibitory control involving the orbitofrontal cortex, the dorsal medial striatum, and the STN. And of course, since we had all these results with STN lesions suggesting that these rats were really highly impulsive, compulsive, then we thought, oh, maybe we have a model of OCD just inducing STN lesions. But interestingly enough, when you apply STN DBS in OCD patients, you have a beneficial effect. So, and STN DBS has also mixed effects on ICD in PD patients. So some patients who were suffering from impulse control disorders can be alleviated when they're subjected to STN DBS, but some get worsened. So it still needs to be assessed further. But it seems like STN is involved in the control of inhibition, and whatever you do, whether it's lesion or DBS, you may actually help some impulse control disorders while you could induce further impairment with a lesion. But now I'm going to move on to the last part of my talk, looking at the role of STN in motivation. As I said, and from the start of the session this afternoon, we've been talking about the reward circuits, and the STN is not even there. So that's a classical view that Marina Wolf had adapted from the very old review by Mogenson, positioning the accumbens at the interface between motivation and action. But actually, there are missing connections on that diagram, of course, and the STN is actually receiving, as I said, direct input from the prefrontal cortex. It's in direct communication with the ventral pallidum, but also receives dopaminergic input from the VTA and the nigra compacta, also controls the dopaminergic neurons, as we've seen this morning with Charles Blaha's talk. So it's nearly almost in the same position as the nucleus accumbens to be an integrative structure to receive all these inputs and be part of this reward circuit and not a simple relay on the behavioral outputs as it used to be on this classical view. And now I'm going to show you results that should convince you that it's really critical for motivation. But what did we know when we started to work on that from the literature regarding STN and motivation? In the rodent, there were our data showing that STN inactivation induces perseveration on the reward magazine in the 5-choice task. In the monkeys, there was one paper by Matsumura reporting changes in the neuronal activity within the STN, while the monkey performing an oculomotor task was expecting its reward at the end of a correct trial. And in the human literature, there were actually, but that's more recent data, showing that an infarct at the level of STN can induce hypersexuality or hyperphagia. And as I said earlier on, there's also this question of gain weight in these PD patients stimulated in the STN. So, and when I said it could be spectacular, we're talking about some patients gain 40 kilos within a few months. So that's really something very interesting. And when you talk with neurologists, they describe you some patients craving for sweet, waking up in the middle of the night and taking the car, driving to get chocolate, and so it can be really serious motivational changes in these patients. But first of all, when we started to look at that, we looked at whether or not when you inactivate the STN, you increase hunger in rats. It's not the case, so I'm not going to show you the results, but we don't induce primary effects on motivation, so they are not more hungry. So no effect on consummatory behavior. Therefore, we looked at conditioned place preference. I'm not going to show you that today, just the progressive ratio schedule of reinforcement, in which we ask the rats to work always more and more to obtain the same rewards. So if I ask you to produce more and more efforts and I don't increase your salary, then you're going to stop after a while and go on strike. So that's what the rats do, and that's what we assess for the level of motivation for the reward. And we started first to look at the effect of STN lesions on food rewards. And what we've observed, in line with what we suspected from the 5-choice task, we've observed that STN lesions, illustrated in orange on these graphs, STN rats are more willing to produce an effort to obtain food reward than the controls. And so that's questions back in the time we did that experiment. We were still focusing on understanding what could happen in Parkinsonian patients subjected to this inhibition of the subthalamic nucleus. And then we thought, if we increase their motivation for food, we are going to increase their motivation for everything, because again, the reward circuit, as I said in the introduction of the session, is supposed to work the same way for whatever reward is on board. So since these patients are taking dopaminergic drugs for their Parkinsonism, they may become addicted to their dopaminergic treatment. So what's going on with motivation for drugs in these rats with STN lesions? And that's how we came to assess the effect of STN lesions on motivation for self-administration of cocaine. And in the progressive ratio schedule of reinforcement, we saw, surprisingly, a decreased motivation. So we see that STN lesions increase motivation for food, but decrease motivation for cocaine. And that's really something we did not expect at all. And I have no explanation for that so far. And it's been a few years now, we try to understand why we have this dissociation. But then the next question was, can we generalize that effect for cocaine to other drugs of abuse? So we've, in collaboration with Martin Cador in Bordeaux, we've looked at the effect of STN lesions on heroin self-administration. And as you can see here, we did FR1 schedule of reinforcement. So each lever press, the rat gets an injection of heroin. FR2, the second lever press, is rewarded, and FR5, they need to press 5 times to obtain an infusion of heroin in their blood. And you see that right from the beginning under FR1 schedule of reinforcement, the lesion blocks the motivation of the animals to take heroin, which of course is confirmed in the progressive ratio schedule of reinforcement. So now we tried nicotine, and surprisingly enough, on nicotine motivation, STN lesions have no effect at all. But you may wonder why, but I mean, the scale is the same, and you see that clearly rats don't seem to like much nicotine, so they don't take that much. Therefore, seeing a decreased motivation to take the drug is really difficult because the level of intake is really low. So there's not even a trend even on the progressive ratio schedule of reinforcement. So you haven't seen, but you can believe me, STN lesions do not affect consummatory processes, but that's really important for our conclusions. But they affect motivation with a dissociation between sucrose and cocaine or heroin by decreasing motivation for the drugs while increasing motivation for sucrose. So therefore, it's a very interesting target for the treatment of addiction, because what you want to do in an addict is decrease the motivation to take the drug, but not decrease motivation for everything, which in brackets excludes the accumbens from the possible target, to my point of view, because you don't want to knock motivation for everything. So with STN, you have a way to dissociate motivation for the drug and for natural activities. But you may argue that's a lesion you're showing us. We don't want to make a lesion in an addict brain. So we have a tool. We have the DBS, and we know how to apply it in the STN. So that was the next step, of course, of our experiments. Better than lesions, could STN DBS be a possible treatment for drug addiction? So first of all, before moving into models of real addiction in the rats, we wanted to make sure that we could replicate the effects I've just shown you with the lesions, applying now bilateral DBS of STN on the progressive ratio schedule of reinforcement for food. So now the control animals are off stimulation. I forgot to mention which parameters we apply. So the stimulation is applied at what we consider high frequency 130 Hertz. That's what is applied in the Parkinsonian human patients. And we apply 60 to 80 microsecond pulse widths, and the intensity is adjusted for each individual, because in the STN, when you apply high frequency stimulation, you induce, if you go high in the intensity, you induce hyperkinetic movement of the contralateral paw. Therefore, you can adjust the intensity for each side of the brain for each individual rat, just going to induce this behavior of this hyperkinetic movement of the paw, and then we go slightly below, just not to be to have any interference of this motor behavior in the task. So having said that, you see that for those animals subjected to the high frequency stimulation, we increase their willingness to work in the progressive ratio for food rewards. And then when we did the same experiment with other rats for working now for cocaine self-administration, then you see that those subjected to the high frequency stimulation decrease their willingness to work for cocaine. So you may wonder that maybe because in the progressive ratio schedule of reinforcement, the session lasts for hours. And so we've stimulated for hours these poor little rats. And maybe we've lesioned the STN by applying such stimulation. One of the advantages of the DBS technique is that it's reversible. So we tested the reversibility of the effect by switching on those animals that were in the off group previously. Now, so they have a longer experience of cocaine self-administration in this task, and now we switch them on, and although they've had more cocaine infusions, there is still a decreased effect by the high-frequency stimulation of STN in these rats. And the reversibility of the effect is interesting for the on group, because now we switch them off and we see whether or not they go back to a higher level of motivation to get the cocaine, and that's what we see. So we have a nice reversible effect. So STN DBS reduces willingness to work for cocaine, but increases that for food in a reversible manner. So now we have an opposite effect induced by STN DBS on motivation for cocaine and sucrose, as we had seen it for lesions. It reduces motivation for the drug, increasing it for food. So not only we have an interesting target for the treatment of addiction, but we have also an interesting tool. And what are the clinical support for our results? We don't have data yet of the effect of STN DBS applied in addicts, just simple addicts to cocaine or any other drug. But we have those Parkinsonian patients addicted to their levodopa treatments. That's called the dopamine dysregulated syndrome. In these patients, we have actually in Marseille a set of patients that we've selected to really assess what would be the effect of the high-frequency stimulation of STN on their addiction to their levodopa treatment. And that's really beneficial because out of 80 PD patients, we had 14 that were diagnosed dopamine dysregulated syndrome. So seriously addicted to their levodopa treatment, and out of these 14, we lost one, of course, in the study, as always with patients, and then 12 totally stopped. They don't even take any levodopa anymore, so that's really, really impressive. So it's a really positive effect, but still, in our rats, you may argue that we were only using conditioned place preference in which they only get exposed to the drug 4 times in their life. That's not really a good model of addiction, although some people have made their career with that. And progressive ratio schedule of reinforcement, the rats in total take only 10 to maximum 15 injections per session per day. So that's not a lot. So what about using a model of more addicted rats? And we decided to use a model developed by Serge Ahmed and George Koob in '98, the escalation of cocaine intake, that allows us to assess the loss of control over the drug intake. So to explain briefly the model, if you leave rats for 1 hour or 2 hours every day in the self-administration chamber, they will always take a constant amount of drug over days. So it's really stable, as you can see here in black. So they're really stable. But if instead of leaving them just this short access to the drug, 1 or 2 hours, you go to a long access, 6 hours per day, they get access to the drug, then you see a progressive intake of the drug, and that is the illustration of the loss of control over the drug intake. So we've replicated that, testing the effect of course, STN lesions first. And then, so we lesion the animal first, and we look at the preventative possible effects of the STN lesions. So they are, in black are the sham animals, and in orange, the STN lesion rats. So during the first block of 5 sessions, that's short access, so they have 2 hours only. And then for the consecutive blocks, it's blocks of 5 days, of 6 hours, but we've illustrated the 2 first hours so that you can compare the level of intake during the first 2 hours. And you see that there is a nice escalation in the sham animals, but in the lesion rats, we don't see the escalation effect. So there is a preventative effect of the lesion on the loss of control over the drug intake. So now we replicated that experiment, now applying DBS of STN. And as you can see, so during the short access, we didn't activate the stimulation. So both groups are off stimulation, and then we started to stimulate during the long access session. And you see that the animal off stimulation shows a nice increase of the intake, so the escalation, while the stimulated animal on in black show no loss of control over the drug intake. And in parallel to that, we also saw a decreased number of perseverations during these sessions, the 2 first hours. So this compulsive lever press during these sessions fits with the effect that I reported already, that STN DBS can actually diminish the compulsive behavior in OCD patients without affecting anxiety, but it acts really on the compulsive aspect of the problem the obsessive compulsive disorders patients have. So now we have a nice descriptive effect. We can prevent loss of control over the drug intake when we manipulate the STN. But why? What's happening? And since we know that STN is also an interesting place where oscillatory activity can be recorded, we decided to use the fact that we implant electrode in the STN to stimulate with high frequency to now record local feed potentials in the STN during the process of escalation. Can we find a marker of the loss of control over the drug intake within the STN? So that was the last recent study we've done. That's Yann Pelou mostly controlling all these experiments. So the behavioral data show you the escalation of the drug intake. So we had self-administration short access sessions here up to 10 days, and then we started the long access for 15 days. Each day, these animals were recorded for half an hour before getting access to the drug, and then after half an hour with access to the cocaine, and then after we would change them in other boxes so that they could complete their 6 hours of administration. And when we record the activity of STN during the start of the session, so they don't get access to the drug yet, we just record the basal activity, then we saw progressively over the course of the escalation, the different steps. So during short access session and then progressively the long access session, we saw increased oscillatory activity in the STN, in the low delta frequencies and in the beta, illustrated here. And that follows really the process of drug escalation in absence of cocaine. So we suggest there are possible markers for loss of control. Of course, you may say, okay, that's preventative effect. So what we really want to see is whether or not you can cure an addict. So now what about if we let all the animals escalate their cocaine intake, and then once they've escalated, we process the STN lesion and we see whether or not they take more or less drug. So that's what we've done on that experiment. And you see that all animals had escalated their level of cocaine intake. Then we did the STN lesion now, and we retested them after the lesion. And you see that there's a decreased number of infusions, although we don't extinguish, of course, self-administration. That would have been fantastic. But you have to bear in mind that these animals, for 6 hours in a box, they have nothing else to do but press on a lever to take some drugs. So they still take a little bit. But that's promising results. And we had actually the same type of results with Yann Pelou did an experiment with alcoholic rats. So he let them escalate their alcohol consumption and perform the lesion. Of course, after the lesion, we had to let them recover. So they had a withdrawal of 20 days before we retested them on their alcohol consumption. And you see that although there was a huge relapse in the controls, then the lesion guys showed a lower consumption of alcohol. So it's promising, not necessarily fantastic and spectacular effect, but there are still promising results. So to conclude, we've seen that STN lesion and DBS prevent the loss of control over cocaine and alcohol intake. During the process of the loss of control, we've seen increased oscillations in the very low frequencies, the delta band 1-4 Hz, also seen in OCD patients. There were also increased oscillations in the beta band that are well known to be observed and described in Parkinsonian patients and OCD patients. And levodopa diminishes these beta oscillations. So therefore, it suggests that the loss of control within the STN mimics an anti-dopaminergic pattern in absence of the drug. It's as if they're going to have the drug, and it's as if in expectation of that, they had a drop in the dopaminergic activity that develops even more during the course of the loss of control over the drug intake. So therefore, applying DBS into the STN could prevent these oscillations to appear and may restore the control over the drug intake, and then may explain why we have this preventative effect. And it's also very interesting, if we think at the possible treatment, that would also be a nice possibility to apply a closed loop stimulation, because as soon as you would detect the oscillatory activity in the STN, then you could start the stimulation and you wouldn't need a constant stimulation to be applied in these patients. Then we've seen that, of course, STN lesion has a potential curative effect on cocaine and alcohol intake, although the intake is not abolished. So the general conclusion, and I don't know how I'm doing with time, but STN plays a role in frontal functions such as attention, the control of inhibition and motivation. It plays a complex role in the reward circuits as a possible gate control function of the hyperdirect pathway, possibly, that needs to be really further addressed. And we've seen that STN oscillations could play a critical role in the control of compulsive consumption of drug and could lead to interesting possibility of closed loop stimulation. And STN DBS may represent an interesting strategy for the treatment of drug addiction. As I said, we've written that review positioning really the STN as the one target that should be aimed for the treatment of addiction with deep brain stimulation. And I finish thanking all the people working with me in Marseille. And we have a translational team, so some people work in the monkeys, some in the Parkinsonian patients, and there are also many collaborations, and I thank you for your attention.